Right, hello everyone. Today I'm going to be talking to you about my PhD project, which is all about building a biologist-centric modeling language for cell-based simulations. Just to give you a bit of a rundown as to who I am, I'm Tom. I'm a PhD student at the Francis Crick Institute, which is a sort of joint biomedical institute with King's College London, UCL and Imperial College London, and I'm supervised by Katie Bentley, who is based at the Crick and is a computational biologist, and co-supervised by Stefan at King's, who some of you may know. So just to give you a brief overview of what I'm going to be talking about, basically, the key gap that my project is trying to address is that simulations are really useful in biology, but require technical skills and understanding that can be rare amongst biologists. And to overcome this problem, we're hoping to create a domain-specific language, a specialized coding language, if you will, to enable biologists to quickly create their own simulations in a reproducible and understandable fashion. But to start us off, I'm afraid I'm going to have to talk a little bit about biology, so bear with me. I'll make it as painless as I can. So our lab at the Crick is interested in a process known as sprouting angiogenesis, which is basically the formation of new blood vessels from existing vasculature. It's a really complicated and highly non-linear process, which makes it really hard to predict how it's going to unfold given a certain set of starting conditions. And it's reliant on a lot of intercellular communication and very intricate shape dynamics. So one example, which you might be able to see in the image I've got on the left, of these really thin and long protrusions, which we call filopodia. And what essentially happens when a tissue becomes low in oxygen, it releases a molecular signal into the environment called vascular endothelial growth factor, or VEGF for short. And in response, cells that belong to blood vessels extend out filopodia towards that, which causes them to sense this signal even more strongly. And once they do so, they become known as active or tip-like cells. And that sort of evidenced by the sort of phenotype they have, with really lots of filopodia and a lot of this molecule called delta-like 4. And when cells become high in delta-like 4, they basically talk to their neighboring cells and say, hey, you need to not do what I'm doing. This causes them to not produce any filopodia, and so they don't sense that signal anymore. And, well, you might be thinking, well, this seems quite nifty, but why would biologists care about this in the first place? You might have guessed, like, growing of blood vessels is really quite important in a lot of developmental and disease conditions. So wound healing being an obvious example, but also in the development of cancer and tumorigenesis, as we call it. Once tumors grow beyond a certain size, they need to have blood vessels basically infuse that tumor so that it can continue to grow. Otherwise, it just sort of necrotizes in the middle and then causes all sorts of nasty stuff to happen. And so if you're a scientist or a clinician who's trying to understand how to treat these diseases, you really need to have a very good understanding of angiogenesis. But because it's a really complicated process, it proved necessary to write computational simulations in order to understand it as well as we can. And as it happens, my supervisor, Katie, wrote during one of her postdocs what's called an agent-based model. And essentially what this is where you represent individual cells, or rather you write the simulation such that you have a collection of computational entities which are able to interact with their environment locally and then en masse produce some emergent behavior. So in this example on the left, we've got a single endothelial cell, which are the types of cells which make up blood vessels. And basically, the cell is sort of represented as a sort of mesh of agents connected together with springs. And it's the sort of combination of agents reacting to the environment locally and the addition of mechanical springs in the model, which allows the cells to do things like move around and to extend filopodia out into the environment. And basically, this turned out to be really key in understanding how angiogenesis unfolds and the sort of requirement for shape for it to occur properly. So I've got a video here on the left showing, what it usually does when you run the simulation. You've got a blood vessel sort of floating around, all the cells extend out Philopodia into the environment, and then the green ones become more active because they're sensing more of the signal, and the purple ones become inactivated. And eventually they sort of settle into this stereotypical, what we call a salt and pepper pattern of stalk and tip cells, i.e. the active and inactive cells. When you somehow prevent the cells from extending out Philopodia properly, which might be a bit difficult to see, basically they become less able to sense this. And once you've got this sort of mechanism in place, you can then begin to do quite interesting things, such as work out the effect of sort of random extension versus not, and things like that, to better understand the dynamics of angiogenesis. And this simulation has been used fairly successfully to predict really non-intuitive things over the past 10 or so years, which required more and more code to be added to the simulation to begin to answer more and more questions, with one example being like the question that we had as to whether the junctions between the cells, i.e. where the cells interface, how does shape changing along those junctions affect the selection process? So you may be thinking, if you're a potential collaborator, well, this sounds great, but the problem is I'm a biologist and I only know Python. Well, then the model is going to prove quite challenging to use, even if you're a really motivated individual, simply because of its sort of like long and varied development history. So some of the problems that began to arose, because of this, are the fact that a lot of the code which controls different features of the simulation are split up and often not fully documented. So, for example, the code which controls Philopodia behavior is sort of split between cell files and individual membrane agent files, which makes it quite hard to work out what's going on. In the model, we talk a lot about technical things which we need to actually run it, and we mix those sort of interchangeably with biological ideas. So in order to actually implement cell shuffling in the simulation, we use a thermodynamical model called the cellular pots model, but a biologist wouldn't know what that is in all likelihood, or if they did, probably wouldn't talk about the problems that they interact with in terms of the cellular pots model. They talk about, I see these cells shuffling when I add this reagent to my dish of cells or something like that. And I sort of touched on this very briefly, but the entire model is not written in a language that's very commonly used amongst biologists. It's written in C++, which, if any of you use, you will agree with me when I say it's not conducive to making changes very quickly, even if you already know C++, and definitely not with a code base that's 10, 15 years old and has had a lot of stuff added to it over time. So how do we begin to address these problems? Well, we can use model-driven engineering, and what sort of gave the impetus for this project in the first place is the fact that a lot of language workbenches have sort of like matured and increased in number over the past few decades. And we think that using a language workbench, which is essentially a tool for creating domain-specific languages, a programming language to specifically target a problem domain, will let us sort of present the simulation in such a way that biologists are able to interact with it a lot more easily. And in fact, this approach has begun to be taken with some other modeling libraries, but a lot of the time these are sort of aimed at more generic problems and don't necessarily talk about the exact shape behaviors that we're interested in to see the really interesting stuff in the biology. And so we thought that making a domain-specific language using a language workbench called JetBrains MPS seemed like a good approach, and I'm going to explain my thinking now. So it was good that Alessio spoke a little bit about projectional editors just now, but in case you're not familiar with MPS, this is essentially what it looks like. It's essentially you've got a list of languages on the left and a user interface on the right. And to sort of explain this, how it works to biologists, I coded a very basic fraction language, which lets you write your own fractions really quickly if you ever felt the need to. But this is sort of the conceit of how it works. So you have your user interface in MPS, but underneath there's a sort of internal representation used by MPS, which is essentially an abstract syntax tree of a model that you're building within this user interface. But the clever thing about MPS is that it uses what's called a projectional editor. And the effect of this is that it allows you to show the same ideas or concepts, such as a fraction, which has a numerator and a denominator, differently depending on whatever context you choose. So for example, on the left, I've got one fraction here where you sort of signify, a numerator and a denominator by having a slash in between the two, like you would usually when you're writing it out. But if I was feeling cruel, I could have them write it in text instead and say divide 1 by 2. And I can switch between those as I please. And this is going to be important later on. And then what you do with MPS is that you can write a set of rules known as generators internally, but are essentially code generators or text generators, which take this internal representation, this tree that you've got building a model with MPS, and then spitting out code which does something which, well, basically whatever you like, or however you decide to have it set up. So my fraction could generate C++ code, which takes in a numerator and a denominator and then does something more interesting. That's a general idea, but if we apply this to the actual simulation, we might have a user interface which talks about things like cell types and signaling molecules. I can have a user interact with MPS to build up a model internally of all the different types of cells and where they're located. I can then get it to generate new C++ code and then have it integrate into the rest of the model to generate hopefully new simulations. And this is essentially what my project is all about. And if I sort of go back to the problems that we had with the model in the first place, we think that using a domain-specific language to sort of address these problems makes sense, because rather than having all the features split up across different documents, we can make DSLs talk about specific features in really sort of regimented and easily recognizable concepts. We can, with our DSL, only have biologists interact with the biology of the model that they're trying to build, and we can write our domain-specific language in whatever language we choose with whatever syntax. It can be plain English, we can have tables, whatever makes sense. So I sort of touched on this briefly, but what sort of made this project a bit different to sort of other DSLs that are used to simulate biology is oftentimes they're sat on top of a more generic modeling library, whereas the problem I have is that the model has been built to do one thing very well, and refactoring this, essentially legacy code base for use with a domain-specific language in the time frame of a PhD is, not necessarily very feasible. So the question becomes, how do I open up the model as much as I can while still preserving all the functionality underneath? So this is essentially my approach, is that I'm sort of interested in different aspects of what the simulation does, and I containerize these different aspects into sub-languages, which is very easy to do with MPS. And I have a sort of overarching simulation container which basically organizes all of these different sub-concepts. And what this lets you do is actually switch them in and out and really sort of promotes a modular approach to building simulations, which I think is quite cool. And the last thing that's worth noting is that rather than generating an entirely new simulation from the model that you build, I have essentially an interstitial layer of C++, which essentially reads like a list of commands, like create cell here and add protein to this cell rather than the other one. And it actually makes it a lot easier to keep track of what you're generating, which I think can be a bit of a stumbling block when you're developing a language like this. So what I ended up with, and I can't get into all of what I've written because it would take far too much time, is essentially 3 different sub-languages for the simulation. And each of these control different aspects of the simulation, like I said. So I've got a world language, which lets you do things like describe how a signaling molecule sort of diffuses in space and describe areas of the world where cells can stick to properly, or, let's call them cell substrates if you prefer. I've got a language that lets you describe the types of cells in the simulation, and also how they're arranged in space. So you can do things like describe a blood vessel cell and then have it arranged in a vessel shape, or have it arranged as a sort of 2D flat monolayer, as we term it. And the last thing, which I'm going to spend a bit of time explaining, is a language which lets you describe essentially communication pathways between cells, like this VEGFR delta-like 4 pathway that I was explaining earlier. So in the signaling language, you can, and this is where I think MPS really shines for stuff like this, is you can define entire reaction pathways within the diagram, and using a projectional editor. But not only that, you can switch between that and this notation here on the bottom, which describes how sets of molecules interact. And then what this language does is, once you've described how different things interact, is that I tell it to generate a set of ODE code, which, if you're not familiar, basically are a set of mathematical equations which describe how the concentrations of these molecules change over time. But this might not be enough if you're a biologist. And one interesting problem that sort of arose as I was developing this is, how do you parameterize these signaling pathways properly? Because you, as a biologist, may know that one thing interacts with another thing, but you may not know how much of a certain molecule is needed, or sort of how much you've got to begin in the first place. So what you can do is you can switch the notation from, just having a single start concentration here, and then do things like specify ranges. And then you can have the DSL automate a sort of interaction with a computing cluster. So when users do this, they can specify, I want to run a grid search using this simulation. I want to find out what levels make sense for me. And then what happens is that MPS is able to also translate this entire simulation into a shell script, as well as produce an executable, and then stick that on a computing cluster and run it. And then, once you do that, you're able to begin to do some more interesting stuff with it. So this is sort of from a while ago, when I was beginning to test the ODEs that I was getting out, or the set of equations that I was getting out from my languages, and just sort of beginning to compare, if I set up grid searches, do I begin to see comparable results? So if I start if I have a setup rather where I have two cells next to each other, and I give one more of this signal which induces angiogenesis, how does that affect the time it takes for cells to decide which one is going to form a new sprout and which one isn't? And you can see that the bigger the difference, the quicker this time takes, as you might intuit. But that's not all we're planning to do, and I'm going to go over this bit very quickly. We do have collaborators who are interested in using simulations produced by the language, and one collaborator in question looks at a organism known as zebrafish, to study how blood vessels form. And what's cool about zebrafish is that they're completely transparent when they're embryos, so you can do quite cool stuff like have all of the blood vessels light up and then just see straight through it with a microscope. But what you might notice is that these vessels form sort of very stereotypical patterns, and they're sort of given a certain amount of space between each blood vessel. And the way that we think this is occurring is that there is a signal in between these regions which basically tell Philopodia not to grow there. And using the DSL, we can begin to describe the spatial arrangement of, these signal blocks relative to a big blood vessel, and see what that does. So I basically do that in the DSL. I can do things like specify where the gradients are found, and then run the simulation and then show what happens over time. So this is just like some ongoing work. So unfortunately, I don't have too much more to show you. But yeah, this is what I wanted to talk about. So to sum up, I hope you agree that simulations can be useful in biology, but they can be quite a sticking point if you're not ready to put the effort in. And to get around this, we think a domain-specific language to speed up the process is a good solution to these problems. And that's all I had, really. Okay, thank you.